Hey there, folks. In science, again, we're looking at cells, and we cannot resist looking at the twin of the endoplasmic reticulum. What I mean by twin, I'm calling it a twin. It's really not a twin. It's just that how would you like to be in the same room as another endoplasmic reticulum? Maybe we just really drive me crazy. But there are two endoplasmic reticulums. One is the rough one. We've talked about that, the one that's involved with protein synthesis. But there's also the smooth endoplasmic reticulum. Just think about this. Think you've got two guys and they're brothers, and one is a roughneck. He's tough. He's hard to deal with. He's rough. He don't take no sense, no nonsense. And then you got the other guy's charming and smooth. Well, that's not really what this is, but it just gives you an idea. They actually do help each other out. They're actually pair. They sort of, I don't say they pair together, but smooth reticulum is something that gives you the it helps you with stuff other than proteins. Let me read this to you real quick. The smooth endoplasmic reticulum is a membranous organelle found in most eukaryotic cells. Oh, I forgot. I got to show this to you real quick. Right here. All right. Now, you remember where that endoplasmic reticulum was right here? Well, it's like right here. There's the endoplasmic reticulum. Well, very close to it is the smooth endoplasmic reticulum. Now, right here, you know, there's the cell. The smooth is right very close to it. It's like it's almost attached to the endoplasmic reticulum. It's very close. Okay, smooth is very close to the endoplasmic reticulum. Smooth is right next to the rough. Anyway, let me continue reading. You find them in eukaryotic cells. Any cell that has a nucleus, right? All right, you got that. So what happens is found in most eukaryotic cells. It is a subset. How do you like that word? Subset. It's a subset. It's something it's in addition to of the endomembrane system of the endoplasmic reticulum. Its main functions are the synthesis. There we go with that word synthesis. We love that word here. Of the lipids, steroid hormones, the detoxification, in other words, they take the toxins out, detoxification of harmful metabolic byproducts, and the storage of metabolism of calcium ions within the cell. The smooth ER is distinguished from the other parts of the endoplasmic reticulum by the absence, now this is always beautiful to have the absence of membrane-bound ribosomes. In other words, those nagging ribosomes, they're not attached. Remember, the rough endoplasmic reticulum has ribosomes attached. That's why they're called rough. Endoplasmic reticulum, the smooth endoplasmic reticulum does not have the ribosomes attached. Therefore, smooth. Okay? All right. It says the smooth ER is distinguished from the other parts of the endoplasmic by the absence of membrane-bound ribosomes. The organelle is also morphologically distinct, safe, often made of tubular structures called cisternae. Smooth ER is prominent in cells of the liver that process harmful chemicals, in cells of the endocrine system, such as those that in the adrenals that produce steroid hormones, and in excitable cells such as neurons and muscle cells that use calcium signaling. Okay, so you got a lot of different functions there. And the smooth ER is primarily composed of three-dimensional polygonal networks of tubules called cisternae. They are about 50 millimeters nms in diameter, not millimeters, micrometer. I can't spell that. Micrometers in diameter in mammals and 30 nms in diameter in yeast. The high curvature of these structures needs to be stabilized by many proteins, including reticulins, DP1, and receptor expression enhancing protons. These proteins either bend the membrane through structural elements that wedge themselves into the lipid bilayer, and they're involved with lipids too, okay, or shape the membrane through oligomerization, and I'm not going to even try to pronounce that again. And the presence of these proteins seems crucial for the existence of tubular cisternae since their suppression or deletion leads to an excess of flattened sac-like structures in ER. Okay, smooth ER is also dynamic structure, with new tubules budding off from the sides of existing structures. With the aid of GTP hydrolysis, some tubule branches can also fuse. And so anyway, the structure of the ER are particularly significant in two types of cells in the human body, muscle cells and neurons. The presence of an extensive ER network along the neuron is closely associated with its interaction with actin and microtubules. So it has a much different purpose than the regular rough ER. Regular rough ER has to do with proteins. This has to do with lipids and steroids and a few other things. So there's a little more involvement, and the ribosomes, remember, are not attached. So that gives you a little more information about this. I was looking to see if there might be anything else I could give you. The smooth ER is important in the synthesis of lipids, such as cholesterol, and phospholipids, which form all the membranes of the organism. In addition, it's important for the synthesis and secretion of steroid hormones. We kind of talked about that, from cholesterol and other lipid precursors. In addition, it's involved in carbohydrate metabolism, for instance, the final reaction of gluconeogenesis, which occurs in the lumen of the smooth ERA, since it contains the enzyme glucose 6 phosphatase. This enzyme catalyzes the production of glucose from glucose 6 phosphate. Anyway, there you have it. Quite a bit of stuff there for the smooth ER, a smooth endoplasmic reticulum, and it's an important part, close to the rough ER, and that should give you a pretty good idea of what you're dealing with. Whew. Are you as exhausted as I am? Anyway, I hope that helps you out. Thanks for watching. Talk to you later.